The sliding filament theory is the process of how your muscles contract. Skeletal muscles, which are connected to bone, allow for body movement. These muscle fibers are made up of myofibrils. Myofibrils contain Z discs, I bands, A bands, M lines, and an H zone. This makes up the sarcomere.
The sarcomere is the structural unit of the myofibril. The sarcomere of the myofibril contains thin filaments called actin and thick filaments called myosin. Each of the actin filaments is attached to a Z line, and the myosin filament lies between the Z lines, which is held in place by a protein called titin. Sarcomeres shorten during a contraction, and so its H zone shrinks. This is done by the myosin heads pulling and sliding the actin filament inward. The actin and myosin remain the same size during the contraction.
The action potential triggers a contraction when transmitted by a somatic neuron and reaches the neuromuscular junction. Acetylcholine is released by the neuron into the synaptic cleft. This triggers the muscle's action potential, which spreads along the muscle fiber's plasma membrane and into the T-tubules. The T-tubules are located in the sarcoplasmic reticulum, which houses calcium ions. When the action potential passes through the T-tubules, it triggers the release of the calcium ions.
The calcium ions diffuse throughout the cytoplasm of the sarcomere. Within the sarcomere is the actin and myosin filaments. The actin is thinner than the myosin, which has knob-shaped heads. Actin has two proteins, troponin and tropomyosin. Troponin is evenly spaced over the actin, and tropomyosin is like a rope that wraps around it. The myosin heads are attached to ADP and inorganic phosphate. After being released from the sarcoplasmic reticulum, calcium ions pair with troponin, which causes them to shift position, revealing the myosin binding sites by pulling the tropomyosin aside. A crossbridge is then formed by the myosin head bonding with the actin. After this bond, inorganic phosphate is released, which triggers the myosin heads to move and pull actin past them, in which ADP is released. The myosin head then binds to ATP, which allows for additional power strokes by making the head release the actin filament. ATP is then hydrolyzed into ADP and inorganic phosphate, which places the myosin heads back into position and starting the process over again.